Welcome STEM enthusiasts!
I am Sophia Lee, an electrical and computer engineering student at Carnegie Mellon University.
And I'm Shaylin Parker, a chemistry student at Carnegie Mellon University. And in Discovering DNA, we are going to introduce you to the exciting world of DNA and genetics.
The first half of this module, we will focus on DNA. We will explore where in the cell the DNA can be found and how to extract that DNA. Then we will learn about mutations in DNA and how to create your own set of DNA instructions to understand the complexities embedded in the blueprint of life.
In the second half of the module, we'll learn how DNA affects what genes organisms have and what those genes look like in our physical appearances. First, we'll introduce the topic of Mendelian genetics and then talk a little bit more about what dominant and recessive alleles are. Then we'll learn how to draw Punnett squares, which are useful tools for predicting what genetics and offspring of two parents might have. Lastly, we'll learn how to trace through pedigrees, which are like family trees, so that we can see how genes have been passed on from generation to generation. At the end of the module, I'm going to put your genetic skills to the test and ask you to solve a murder mystery. We hope you enjoy this module we put together. And remember, have fun!
Welcome back, STEM enthusiasts! Today we are going to be creating a basic model of the cell, the smallest structural and functional unit of an organism. We will be discussing the structure and function of the cell wall, cell membrane, and finally the cell nucleus. So, the cell is the tiniest functional living unit in the world. What does that mean? Think about what you need to be a person. You need a heart to pump blood, you need a digestive system to process food, skin to keep everything inside, and all your other organs. A cell has all the working components within it. Each component is called an organelle, kind of like your own organs, which all work together to make the body function. The first organelle we're going to talk about is the cell wall. Cell walls are only in plant cells, which help to create a more defined shape to the cell. We will be using a Tupperware container to represent this. Any type of container, Tupperware or otherwise, will work. It can have different pockets within it, or not be clear, or be something cardboard like a tissue box or shoe box, or even a mac and cheese container that has an opening you cut out. The reason I use a Tupperware container for our model of the cell wall is because it is both rigid and water resistant, because cell walls are also water resistant. Now I'm going to decorate my container a bit with things to help me remember the parts of the cell wall. Be right back. And we're back. See, I decorated it with just the cell wall, a microscopic picture of a cell wall, and a diagram of cell walls. Now that we have our model, can you think of an organism that has a feature that acts similar to a cell wall? I'll give you some time. One example I've decided to go with is a crab shell. A crab shell protects the crab while maintaining its shape. The second organelle we're going to talk about is the cell membrane. The cell membrane is made up of a lipid bilayer. The outside of each lipid molecule repels water while the inside attracts it. But these two are put together, kind of like tape, where one side is sticky and one side is not. But if you take two together, you put them together, then both sides are able to repel water. And that is how cell membranes work. The outside both repel water to keep water in the cell and outside of the cell. For our model of the cell membrane, I will use a gallon bag. Similar reasons to the Tupperware container, but a cell membrane is more malleable and less rigid, though it also does keep water in and out. Now I'm going to decorate this as well. And so this is my decorated cell membrane with this picture that I've been using as our model on screen, and then a couple pictures of a lipid bilayer so you can see and remember what it looks like. I'm just going to plop it inside the cell wall because that's how the cell works, how it's formed. Now that we have our model of the cell membrane, can you come up with some organism that has structures like it? For me, I chose the human and our skin. Our skin helps decide what comes in and out of our bodies, what it absorbs. It controls what stays inside of us and what stays outside of it. The third organelle we're going to talk about is the nucleus. The nucleus contains DNA, which are the blueprints for life, or instructions telling us how to function. The nucleus is especially important because it is used to protect the DNA from becoming damaged. Later on in this module, we'll see what can happen if DNA is damaged. I am just going to use a regular old sandwich bag for this, but remember, you don't have to use these materials if you don't have them. This could easily be a grocery bag, and this could be some other type of bag, or even a blow-up ball or balloon. Anything that you have laying around that's malleable and that will help you remember what each of these do will be great. Now I'm just going to decorate this bag and I'll be right back. And here's my decorated bag. So it has the little model of the nucleus that is also up in this corner. Then it has a picture of what goes on inside the nucleus, and then a picture of what the nucleus looks like under a microscope. I'm also going to put in this picture of DNA to remind us that DNA goes inside the nucleus. And put it in my model. Can you come up with an organism that has a structure similar to the cell nucleus? One structure I've come up with, though it doesn't exactly fit, is your skull. Your skull helps protect your brain, and your brain is the one giving directions to the rest of your body. Kind of like how the cell nucleus protects your DNA, which gives instructions to the rest of the cell. And congratulations! You have just created the most basic construction of the cell. One thing we do have to discuss, though, is how accurate is this construction? Well, considering it's made of materials laying around your house, I wouldn't say too accurate. Imagine that we just use skin, bones, and your brain to help represent the human body. Would it represent the main structure of human? Yeah, kind of. But would it be enough to create a functioning human? Not exactly. Hope you enjoyed, and remember, have fun.
This activity, I'm going to challenge you to create 2 to 3 more organelles for your own model of the cell. Some examples I'm going to use are Golgi bodies, which I'm going to use straws to represent. Golgi bodies receive proteins and redistribute them throughout the cell, and they are a tube-like structure. Rough ER, or endoplasmic reticulum, are represented by crumpled up gift paper. I'm doing this because the rough ER packages proteins and it looks like a bunch of flattened tubes. Some other choices could be mitochondria represented by beans or lysosomes represented by Lysol wipes. The next time a teacher asks you to create a model of a cell, you don't have to spend hours with clay trying to shape everything precisely. Instead, just go around your house picking up materials and make a simple model of a cell. In summary, the vocab we learned was organelle, cell wall, cell membrane, lipid bilayer, and nucleus. An organelle is a structure within a cell designed to do a specific task. The cell wall helps keep the cell shape and is an added layer of protection. Shell for protection. The cell membrane is made up of a lipid bilayer and determines what substances enter or leave this cell. Skin for containing and protection. The nucleus holds the DNA of the cell. Skull for containing the brain for telling the body what to do. Next up, extracting DNA. Caution, mess alert. We will be using liquids that can spill and/or create a mess. Ask an adult if where you're working is okay. Having paper towels at hand or working on top of a garbage bag or cardboard can help lower the potential mess.
Welcome back, STEM enthusiasts! Today we are going to be extracting DNA from cells. Last time we learned where in the cell the DNA is stored, and we are going to be using that knowledge to extract the DNA today. The first step in extracting DNA is creating our extraction liquid using water, salt, and detergent. Now we're supposed to put about half a teaspoon of salt, just gonna guesstimate. About a third of a cup of water. And 1 tablespoon of dish detergent. Now I'm going to stir this together. You want to try to make sure there's no bubbles forming, but that everything is mixed well. Our next step is going to be to put our filter. I'm using a coffee filter. You could use a cheesecloth or some other filter over a cup. Okay, it looks like all our preparation is done. Time to start extracting DNA. Today, I'm going to be extracting DNA from a banana. I can use about a third of a banana or 3 strawberries, or there's many other conversions you can look up online. Okay, now that my banana is in the bag, I'm just going to mash it up until it's a tiny little pulp. Based on our cell model, what do you think the purpose of mashing up your fruit is? Mashing up your fruit helps get rid of the cell wall. So I'm gonna get our model and remove the cell wall. So right now, our model only has the cell membrane and the cell nucleus protecting the DNA. Once your fruit is significantly mashed, you can add the extraction liquid. I'm going to add about 3 tablespoons of my extraction liquid. I'm going to reseal the bag, trying to get as much air out of it as possible. And then mash up my banana and extraction liquid mixture, trying to keep all the bubbles to a minimum. While I'm mashing this up, can you think of a reason why we use detergent or dish soap in our extraction liquid? Well, if you thought about the cell membrane, you would be correct. As you learned last time, the cell membrane is made up of a lipid bilayer. Lipid, of course, means fat. And as you've probably noticed, whenever you have used butter or oil, soap helps a lot in breaking down that oil and fat. It does the same purpose within these bananas, breaking down all the cell membranes until we're left with our cell nucleus. So I mixed the detergent and banana together for about a minute, and now I'm going to take my cup with the cheesecloth or coffee filter over it and slowly try to pour this mixture into it.
While the soap breaks down the lipid bilayers, the filter helps to filter out all the membranes and other large organelle structures. As you can see, I have to wait a bit for the mixture to drain through the filter. I just set a timer and came back every 5 minutes to check the progress. I also tapped the top of the filter lightly to try to get more liquid out. Be careful, you don't want to break your filter. I used a half teaspoon measuring device to transfer my liquid from the cup to these containers. Any clear container will work. I could have kept the liquid in the cup or used the rectangular box holding these tubes. These tubes actually came from a bouquet of flowers. I'm also using 70% isopropyl alcohol that has been put in the freezer. At least 70% alcohol is recommended, though a higher content will yield you more DNA. I'm using these identical tubes to carry out a little experiment. I want to see if the ratio of DNA liquid to isopropyl alcohol affects the amount of DNA I get. On the left, I filled up less of the tube with DNA liquid, and to the right, I filled up more. Now it is time to extract the DNA. There are many recommended ways to extract the DNA. You can use something wooden like a toothpick or chopstick, or use something metal like a paperclip or pin. You can see me trying to scoop up the DNA from one tube. I was using a chopstick because it was what I found first, but it looks like maybe a smaller implement may have been better at extracting the DNA. Now you're going to get your first look at DNA. The challenge of this activity is to make a hypothesis, test it, and make a conclusion. For my hypothesis, I said more DNA liquid to isopropyl alcohol will give me more DNA. Then I tested it. I used different tubes and filled them up with different proportions of the DNA liquid, then filled them up to about the same mark with isopropyl alcohol. Lastly, I made a conclusion. My hypothesis was disproved because if you didn't have enough alcohol to draw out the DNA, you won't get as much DNA. Now I'm going to challenge you to come up with a hypothesis, test it, and come to a conclusion. Different possible tests can include using different fruit, measuring out your ingredients carefully and changing only one variable, like the amount of soap, water, and salt you put into the extraction liquid, or how much extraction liquid you use compared to the fruit. Or you can put some alcohol in the freezer and leave some at room temperature and see which one extracts the most DNA. Or you could design your own test. Think of all the steps we went through to get to this point. We had to mash up the fruit to destroy the cell walls, then use an extraction liquid to break down the lipid bilayers in the cell membrane and cell nucleus, while the salt helped the DNA strands to clump together. After, we used a filter to get rid of the larger organelles, and lastly, we used the isopropyl alcohol to draw out the DNA from the rest of the liquid. Before we go too in-depth with DNA, we should first define what we are learning. DNA, or deoxyribonucleic acid, is a complex biological molecule that carries hereditary information in many organisms. In other words, It is kind of like our own brains, which carry all of the information our body needs to survive. Even if we don't consciously use all the information, it still keeps our heart beating and our lungs pumping. Nucleotide consists of a nucleoside linked to a phosphate group. Nucleotides form the basic structural unit of nucleic acids such as DNA. Today we're going to talk about 4 nucleotides in particular: adenine, thymine, cytosine, and guanine. The last vocab word we're going to learn in this activity is base pair. A base pair is 2 nucleotides linked strongly together by hydrogen bonds. To help keep all this new vocabulary straight, let's look at an analogy. It is always said that DNA is the blueprint of life. Well, we're going to compare what we just learned to our written language. Nucleotides can be seen as letters, which are small and don't do much on their own, but are needed if we are to continue any further with written language. Base pairs can be compared to words, though a better analogy would be to compare them to phonemes, the different sounds we have in language. However, most people don't know what phonemes are, so it's not exactly helpful. Words are a little more substantial, but you need at least a couple of them to tell a story. However, if you put lots of base pairs together, you get a strand of DNA. A strand of DNA can be compared to a paragraph, which tells a good portion of a story. Later in this module, we'll see what we can compare an essay to. Remember how we extracted DNA in the last experiment? Now we get to see what that DNA looks like up close and how it is formed. Creating the template. If you have a printer, you can print out a template of the DNA helix. Just look it up online. But I will be going through the steps to make your own template. First, take a piece of blank paper and fold it hot dog style. Then fold it about 3 times, ending up with 16 rectangles, 8 on each side. The second step is to draw the semicircular shapes and the arrow shapes along the middle crease, as seen in this video. Number 3. This can be swapped with number 4, but I just preferred to do it this way. So you're going to take your 4 colored pencils and color each different shape a different color. For me, I decided to go with red or pink, green, yellow, and blue as my colors. I labeled each color adenine, thymine, guanine, or cytosine. These each correspond to 1 color and 1 shape. After coloring and labeling each of your 4 colors / shapes / nucleotides, you will cut them out and set them in their own piles. Let's talk a bit about base pairs. Base pairs refer to how the 4 nucleotides can always be arranged into the same 2 couples. Adenine will always go with thymine, and cytosine will always go with guanine. This grouping happens similarly to how we made our DNA model. The 4 nucleotides each have a specific structure which only allows for the previous couples. I like to remember how the nucleotides work together with the following mnemonic device: all tigers can growl. Because it's true. Now, let's pretend one protein that the DNA codes for has the base pairs on one helix of A, C, G, T, T, A, C, C. The first step to creating a DNA double helix is to lay down each letter so the flat edges are on the same side. I colored both sides of the paper to help me see which piece of paper was which nucleotide. Let's see. First comes A, so we pick out adenine and set it down. Then comes C, so we pick up cytosine and put it down. And so on and so on. We have guanine, thymine, thymine, adenosine, cosine, and cosine. So, once we have all the nucleotides lined up in one line, we can take each nucleotide's counterpart and match them up. Now we have a double helix strand of DNA. Let's see if you can create your own based on what you just learned. The next strand of DNA is GCAAATGG. Good luck! If you're looking for more of a challenge, look up transcription. Transcription is what happens when DNA has to replicate, and see if you can replicate that with what we just built. Or you could design your own test. DNA give instructions. As we have discussed multiple times previously, DNA is the blueprint for life. Today, we will be creating our own set of instructions for a basic task, and we'll get to see what happens to these instructions when they encounter mutations. Before we go on, we should learn some new vocabulary words. Earlier, we learned about nucleotides, base pairs, DNA strands, and we hinted at what a gene was. Well, now we're going to define it. A gene is a specific sequence of nucleotides that has the ability to completely or partially control the expression of one or more traits in every type of living organism. An allele is a specific variation of a gene, like you can have an allele for brown eyes and an allele for blue eyes, but when someone looks at you, they will see your trait of brown eyes. Each human has two alleles of a specific gene. A mutation is an alteration in the nucleotide sequence of the genome of an organism. Mutations can go from being banal and ineffective to causing cancer. Here we have a picture of how we can construct the DNA we learned about in the last two activities to the model of the cell from the first activity. DNA is stored in chromosomes. Chromosomes keep the DNA from becoming tangled and damaged. Chromosomes are also what scientists look at when they are studying DNA and your alleles. See where the two alleles come into play? These will be described in greater detail later in the module. There are three mutations in DNA we're going to talk about today: substitution, deletion, and insertion. Substitution is when one base pair is switched. Deletion and insertion both change the number of DNA bases. Deletion does this by removing a piece of DNA, which can be as small as a few base pairs within a gene, or large, like removing an entire gene or neighboring genes. Insertion changes the number of bases in a gene by adding a piece of DNA. As stated previously, mutations can either have no effect, where no physical expression or change to the protein is visible on the gene, or they can have effect, where a trait is altered. For my instructions, I will be focusing on how to brush your teeth. You should try to keep your instructions to simple sentences because DNA instructions are basic, and the only way for a cell to know what to do is if we give them simple directions. I cheated a bit on lines 10 and 11 because I didn't want these to get too long. The first 5 instructions are grab your toothbrush, turn on the faucet, pick up your toothpaste, open the lid of toothpaste, and squeeze toothpaste tube over toothbrush. The next few lines are put toothbrush under the water, turn off the faucet, open mouth, put toothbrush in mouth, then brush top teeth, front and back and ends, each for about 15 to 25 seconds. Number 11 is brush bottom teeth, front and back and ends, each for about 15 to 25 seconds. Line 12, brush teeth and gums. Line 13, take toothbrush out of mouth. Lines 14 through 18, turn on faucet, put toothbrush under water and clean off toothpaste. Put toothbrush back on its container. Pick up a cup, fill with water. And the last few instructions are bring cup to mouth and rinse mouth with water. Bring cup back down, spit out water, and turn off faucet. And those are the instructions for brushing your teeth. Now let's look through possible mutations to this DNA. We'll start with substitution. If we substitute the word toothpaste with soap in line 15, someone might not even notice the error, so this change will have little effect. A larger outcome may come from substituting the word toothbrush with hairbrush in line 1. This would be a much larger cause for concern for most people. For deletion, we could delete line 7, which tells us to turn off the faucet. This is actually normal to keep the water running for some people who brush their teeth. A large effect would be deleting line 8. Without opening your mouth, your teeth will never be brushed. For insertion, we could put the line, put toothbrush under water after line 2. This is just a little extra effort and doesn't change the outcome. However, a very large change could be to insert go eat a sandwich after line 10, or something helpful like adding flossing instructions after brushing your teeth. See, not all mutations can be bad. Mutating our instructions can be a bit different than what happens in actual DNA mutations. For DNA mutations, insertion and deletion have the opportunity to have much larger outcomes than substitution may have. Imagine you wrote an essay. It's a great, A-worthy piece of work. Now, you change some word in the essay. You may still get an A on the assignment pretty easily. Now, imagine you added something to the essay. This could make the essay better, keeping it at an A, or maybe it's a paragraph about hamburgers in the middle of an essay about an English poet. Less good, maybe dropping your grade to B or C. Finally, maybe you delete something from your essay. Deletion causes a lot of damage. For example, if your entire essay got deleted, then you'd end up with an F. Now I challenge you to create your own set of instructions. Try to make one with at least 10 lines, but you don't have to go overboard with like 40 steps. Then, come up with at least 4 different mutations to your set of instructions. Some ideas for instructions could be making a sandwich, washing your hair, putting on a pair of sneakers, or folding a t-shirt. To recap, mutations can be as small as 1 nucleotide or tens of thousands of nucleotides long, visible on a chromosome. Every mutation has the possibility to cause a protein to not function properly. This may cause an allele within a gene to change. If an allele affects the outward appearance of the organism, then you will be able to see the different trait. Mutations are completely random and can either be harmful, beneficial, or have no effect at all. Finally, substitution, deletion, and insertion are 3 possible mutations.
Hi again, my name is Shaylin, and I'm going to be taking over the second half of this Discovering DNA module. And in this half, we're going to talk more about the physical expressions of DNA and genetics. Genetics is the study of genes, or the sequences of nucleotides in our DNA. Before we get started with this half of the module, take a look over some of these vocabulary words that you should be familiar with by now. We'll be using them throughout the video, so if you need to pause so you can read through these definitions one more time, go ahead. We already know that DNA is the blueprint for life and is responsible for most of the structural and functional differences between organisms. For example, my DNA is vastly different from that of a bacteria, and it's clear to see that we are two very, very different organisms. However, DNA is also responsible for some of the subtle differences between organisms of the same species. These more nuanced features make up a very small part of the genome, but contribute to human, plant, and animal individuality. The genome is just all of the genetic information for a certain organism. It's the reason why you see small differences in a litter of kittens that come from the same parents, and the reason why children look similar to their parents but not identical, and also why siblings don't look exactly identical to one another, except for the case of identical twins. One of the first scientists to do major research in this area was Gregor Mendel. Mendel wondered why he observed so many physical differences between pea plants. Sure, functionally, the pea plants were doing the same thing. They were all growing, flowering, producing seeds when pollinated. But he was more curious about some of the minute differences between them. Mendel noticed that some of the pea plants had purple flowers while other ones had white flowers. Some of them were tall while others were quite short, and some of them produced round seeds while others produced wrinkled seeds. These were only some of the differences that Mendel noticed among the pea plants, and this made him curious. Not only did he notice these differences between one generation of pea plants, but he also saw genetic differences between parents and children. For example, he would observe 2 pea plants with purple flowers, but their offspring would have white flowers. And like I mentioned before, he realized that not all offspring from the same 2 parents were exactly the same. To answer some of his questions, Mendel decided to do vast research on these pea plants, and he actually bred thousands and thousands of them across many generations. What he would do was take a small paintbrush and cross-pollinate the plants. So he would take the pollen from one pea plant and transfer it to another pea plant in order to fertilize, so then it could go off and produce seeds, which would turn into offspring that he could track the physical traits of. These are some examples of the physical traits that Mendel was tracking in his pea plants. It is important to note that this act of cross-pollination is an example of sexual reproduction. We'll talk more about sexual versus asexual reproduction a little bit later, but I just wanted to point it out. Through his years of research, Mendel was able to come up with 3 fundamental laws of inheritance. Inheritance is the process of children acquiring traits from their parents. The first law is the law of segregation, which states that for each trait, a parent will only pass on one of its 2 alleles. The second law is the law of independent assortment. This just means that the passing on of one trait does not affect the passing on of another trait. For example, if a parent passes on a purple flower color allele, this will not have any effect on whether or not that same parent passes on a round seed shape allele or a wrinkled seed shape allele. The third law is the law of dominance, which states that the dominant allele will overpower or dominate the recessive allele.
These 3 laws of inheritance are fundamental to our understanding of genetics today.
Before we go on with Mendel's laws, let's pause and take a look at some of the vocabulary that I just threw at you. Dominant and recessive. What are dominant and recessive alleles? For most of the traits we'll be looking at today, there are 2 possible alleles, a dominant allele and a recessive allele. As the name implies, if you put a dominant allele and a recessive allele together, the dominant one will overtake the recessive and it will look like the recessive allele was not there at all. For example, if we were looking at some alleles for eye color, the dominant allele codes for brown eyes while the recessive allele codes for blue eyes.
It's important to note that we always indicate a dominant allele with an uppercase letter, and we always indicate a recessive allele with a lowercase letter.
If you put a dominant allele and a recessive allele together, that brown eyes dominant allele would totally cover up the blue eyes allele, and you would get a baby that had brown eyes. And you would have no evidence of that baby having a blue eye allele at all, unless you were to look a little bit deeper and look at its genotype.
Another quick thing to notice here is that whenever we write a dominant allele and a recessive allele together, you always put the dominant allele or the capitalized letter first.
Sometimes people refer to dominant and recessive traits, and this just means the physical expression of the dominant allele, brown eyes, or the physical expression of the recessive allele, blue eyes.
When we're talking about genotypes, or the specific alleles that an organism has, it's important to note that genotypes fall into 3 different categories. So we've got some new vocabulary to help us with that categorization. The first term to know is heterozygous, which means that an organism has 2 different alleles, in this case, 1 dominant and 1 recessive allele. The opposite of that term is homozygous, which means that an organism has 2 of the same allele. It can either have 2 dominant alleles or 2 recessive alleles. The little table at the bottom of this slide is an example of all the different allele pairings and their corresponding genotype categories. The table also describes what phenotypes are a result of the genotypes, or what physical expressions occur because of an organism's genotype.
To get more familiar with the concept of dominant and recessive alleles, complete the Create a Dog Lab, where you will let chance decide various physical traits for a dog, and you will create a beautiful dog Frankenstein, maybe a new species.
To get started on this activity, you will need the Create a Dog Lab worksheet, a pen or a pencil, a coin, and something to color with. I have crayons. For each trait on the worksheet, you're going to flip a coin 2 times. Mark down the results of those flips in order to get your allele pair. Heads is the dominant allele, and tails is the recessive allele. I flipped tails for each of the alleles for the body size trait. Therefore, my dog will have 2 recessive alleles for this trait. Then I fill in the genotype category, which is homozygous recessive, and I fill in what phenotype my dog will have based on the descriptors in the trait box. Continue this process to figure out every trait for your dog. When you've finished collecting all of your traits, draw a picture of what your Franken dog might look like. Go ahead and pause the video and get started on the Create a Dog Lab.
Here's what my dog looks like on the side of the screen. Take a moment and consider how similar or different it is to yours. I've done a little bit of math here, and it turns out that just looking at these 9 traits, there are a possibility of 512 different dogs that you could get out of this lab with all different traits. Think about how this applies to people. Yes, genes are the reason that people can look very different from one another, but interestingly enough, 2 humans, no matter how different they look, share 99.9% of the same exact DNA. This means that only a 0.1% difference in DNA is contributing to why we physically look so different from some of the people around us. It is amazing to think that even though we don't all look the same and that we all come from different backgrounds and have different family trees, we still are much more alike than we realize. The proof is in the DNA.
You might be wondering more about how these 2 alleles are passed on from parents to offspring. I promised you a small segment on asexual and sexual reproduction, and this should answer that question a little bit more for you. For species that engage in sexual reproduction, one allele is passed on from each parent, one from a mom and one from a dad. The way this works is that mom and dad's DNA combine in order to make a new set of DNA for their offspring that is a combination of both mom and dad's traits. Some organisms do not have to engage in sexual reproduction in order to have offspring. This is called asexual reproduction. And essentially, these organisms are able to make a copy of their own DNA and then split their cells in half. So effectively, one cell doubles into two identical cells. In this case, there is no combination of DNA that's occurring. The DNA of the new cell is exactly the same as the DNA from the parent cell, assuming that no mutations occurred in the replication process. Cells that copy their own DNA and then divide in half are going through a biological process called mitosis. On the other hand, cells that combine their DNA in order to make a daughter cell are going through a biological process called meiosis. And I've drawn a very, very simplified version of meiosis on the board, but if you want to do more research, there are plenty of resources out there that can give you the step-by-steps. Let's highlight some of the key differences between asexual and sexual reproduction. Asexual reproduction consists of one parent, while sexual reproduction consists of two parents. Asexual reproduction results in offspring that look identical to parents, while in sexual reproduction, the offspring produced look similar to, but not identical to the parents. The offspring in asexual reproduction have the exact same DNA as the parents, which explains why they look identical to their parents. And on the other hand, with sexual reproduction, the offspring have a combination of the parents' DNA, which is why they look similar to each of their parents, but not exactly the same. For the rest of the video today, we will be exclusively focusing on sexual reproduction.
Let's look back at Mendel's 3 laws of inheritance.
We've got the law of segregation, which says that each parent passes on only one allele for each trait. We've got the law of independent assortment, which says that pairs of alleles are passed on independently from one another, meaning that the passing on of one allele won't affect the passing on of another. And we've got the law of dominance, which states that a dominant allele will dominate or mask a recessive allele if they are both present.
In this next activity, I am going to give you a piece of inheritance data that Mendel collected from his pea plants, and I want you to pick the law of inheritance that best describes this phenomena. Keep in mind that sometimes there is overlap in what laws explain what phenomena.
Read through this scenario and decide which law of inheritance is responsible for this data. The answer here is the law of independent assortment. The law of independent assortment states that the passing on of one allele is independent of the passing on for another allele. This means that even though parent 1 and the offspring both have wrinkled seeds, the passing on of the wrinkled seed allele has nothing to do with the flower color allele. So we can't assume that just because both have wrinkled seeds, they would both have purple flowers as well. Here's the data from scenario 2. The reason that there is 0 chance of their offspring having green pods is due to the law of dominance. Yellow pods are recessive traits, and if both parents have yellow pods, they must each have 2 recessive alleles. That means that only recessive alleles could be passed on to the offspring. Since the offspring has no dominant alleles whatsoever, that means that the recessive allele would not be masked, and we would see yellow pods. Try out scenario 3. This phenomenon is due to the law of segregation. If parent 1 is homozygous dominant, then it has 2 dominant alleles. Parent 2 is homozygous recessive, so it has 2 recessive alleles. According to the law of segregation, each parent will only pass on 1 allele for the pod-shaped trait. Parent 1 will pass on one of its 2 dominant alleles, and parent 2 will pass on one of its 2 recessive alleles. This would lead to an offspring with a heterozygous genotype, which is not identical to either of the parents. Since the offspring has a heterozygous genotype, and we see a dominant allele in there, that means that the offspring would show the dominant phenotype, which is inflated pods. This is the last of the laws of inheritance practice problems. See if you can figure it out. The answer to this scenario is actually the law of segregation and the law of dominance. If the offspring has white flowers, then it must carry 2 recessive alleles. The offspring gets 1 allele from each parent according to the law of segregation, which means that both parent 1 and parent 2 must have had the recessive allele. However, since we see the parents displaying the dominant purple flower phenotype, they must also have the dominant allele, which makes them heterozygous. The law of segregation explains why the offspring gets 1 allele from each parent, and the law of dominance explains why the parents had purple flowers, but the offspring had white flowers.
These 3 concepts, known as Mendelian genetics, are incredibly important when making predictions about what offspring will look like as well. For example, if 2 parents have brown eyes, scientists are interested in knowing the probability of their child having blue eyes. In order to do this, scientists have to know the genotype of each parent, or the different allele combinations they have. Then, they can come up with different combinations of the parents' alleles in order to figure out what physical trait the child might have, or what phenotype they might have. Rather than doing all of this math in his head, a scientist named Reginald Punnett developed a tool called a Punnett square, which is able to track all of this data that we talked about. With a Punnett square, scientists could easily see every combination of alleles from the parents and easily determine the probabilities of a child having a certain phenotype.
In this tutorial, I'll be teaching you how to draw and use a Punnett square, and how to calculate some basic probabilities about genotypes and phenotypes. We'll keep talking in terms of pea plants because that's what we're most familiar with. Let's say that gene P controls the flower color trait. The dominant allele, which is indicated with a uppercase P, codes for purple flowers. The recessive allele, which is indicated with a lowercase p, codes for white flowers. Like I mentioned before, we need to know the genotypes of each of the parents in order to do the Punnett square. So for parent 1, in this example, we've got a heterozygous genotype, and since we know the details about dominant and recessive alleles for this trait, we know that a heterozygous genotype will code for purple flowers in the phenotype. Parent 2 has a homozygous recessive genotype, which means that its phenotype must be white flowers because we've got those 2 recessive alleles and no dominant alleles to mask anything. Based on this data we have for the parents, we want to find out what are the possible genotypes and phenotypes for an offspring of these 2 parents. This is where a Punnett square will come in handy. I like to write the genotypes of each of the parents in the top left corner when I'm doing a problem like this, just so I have it for reference. Now I'm actually going to start drawing the Punnett square, and as you might imagine, first you start out with drawing a square, and then you want to split that square into 4 equal sections. Across the top of the Punnett square, I'm writing the 2 alleles from parent 1's genotype, and down the side of the Punnett square, I'm going to write the 2 alleles from parent 2's genotype. Next, we're going to start crossing parent 1's alleles with parent 2's alleles in order to figure out every possible genotype that an offspring could have. You can see that I'm bringing down parent 1's allele into the top left box and bringing across parent 2's allele into the top left box. So we're essentially making a little intersection point and then writing both of the alleles that cross to make that intersection. I do the same with the bottom left square, bringing down parent 1's dominant allele and bringing across parent 2's second recessive allele, and I continue on with the top right box and the bottom right box. Doing each of these crosses just shows me every possible allele combination if you pick one allele from parent 1 and one allele from parent 2. So, all of these genotypes I've written in the Punnett square are every possible genotype of any offspring of these two parents. Just to clarify, we've got 2 instances of the heterozygous genotype and 2 instances of the homozygous recessive genotype. We don't need the genotypes of the parents anymore since we've already made the Punnett square, so I'm replacing that information with the details of the 2 genotype categories we're seeing in the Punnett square. One of them being heterozygous, which will code for purple flowers, and the other being a homozygous recessive genotype, which will code for white flowers. Now we can start answering some probability questions regarding the offspring's possible genotype or phenotype. In this case, we're asking what are the chances of the offspring having purple flowers? We can see from the Punnett square that we have 4 possible allele combinations. 2 being a heterozygous genotype and 2 being a homozygous recessive genotype. I'm considering these 4 different genotypes in the Punnett square because technically, if we're looking at just the heterozygous, for example, the first heterozygous genotype in the top left is made up of parent 1's dominant allele and parent 2's first recessive allele. And if we look in the square below it, at that bottom left square, the heterozygous genotype is made up of parent 1's dominant allele, but parent 2's second recessive allele. So that's why we're calling it 4 possible genotype outcomes, rather than just the 2 genotype categories of heterozygous and homozygous recessive. What we care about for this question are the 2 genotypes in the Punnett square that will actually give us purple flowers, and we'll call these the successful genotypes because they result in the desired trait, purple flowers. So if we put these 2 successful cases over the 4 cases altogether, the 4 possibilities, we get 2 over 4, or a 50% chance of the offspring having purple flowers. You can visually see this in the Punnett square too, because if you split it in half, you'll have half of the genotypes giving you purple flowers and the other half giving you white flowers. Another possible question we could get here is what are the chances of an offspring having a homozygous recessive genotype? And I'm going to follow a very similar process to what I did before. We can see from the square there are 4 possible genotypes altogether, and only 2 of them are successful or give us that homozygous recessive genotype. Once again, that gives us 2 successes over 4 cases altogether, which is a 50% chance of homozygous recessive. Sorry, that text got cut off a little bit. Now, if we change up the question slightly and instead ask what are the chances of a homozygous dominant genotype, our math looks a little bit different. We've still got 4 possible genotypes, but when we look to see if there are any successes in the Punnett square, there is actually no instance of a homozygous dominant genotype, no instance of those 2 dominant alleles together. Therefore, we've got 0 successes out of 4 cases altogether, which gives us a 0% chance of the offspring having a homozygous dominant genotype. Now that you've had this introduction to Punnett squares, I'd like you to pause the video on the next slide where I show you a few practice problems that you can try on your own. Once you've tried all the problems, come back and I will go over the solutions and how I got to those answers. You can use a piece of scrap paper to draw your Punnett squares and do your calculations. Hopefully you've gone ahead and attempted those practice problems on your own, because now I'm going to start going over them. So in problem 1, we are told that parent 1 has a homozygous dominant genotype and parent 2 has a homozygous recessive genotype. Now, rather than having those large words there, I find it easier to right away just convert those into genotypes. So we know for homozygous dominant, we've got those 2 dominant alleles, 2 uppercase P's, and for homozygous recessive, we've got the opposite, those 2 recessive alleles or 2 lowercase p's. Once again, I'm going to draw out a Punnett square in order to answer the follow-up questions. So I draw that square, split it into 4 sections, and write parent 1's alleles across the top and parent 2's alleles down the side. Then I begin my crossing procedures that I talked about in the tutorial, where I bring down the allele from parent 1 and bring across the allele from parent 2 so that they intersect in one of the boxes in the Punnett squares. I then write out these alleles and I can see every possible genotype that an offspring could have from these parents. So question 1a asks me what the chances are of white flowers. In order to get an offspring with white flowers, we know we need that homozygous recessive phenotype, those 2 lowercase p's. If we look at the Punnett square, we have no instances of that, or no successful cases. So those 0 successes over the 4 total genotypes in the Punnett square gives us a 0% chance of the offspring having white flowers. Question 1b asks what are the chances of purple flowers, and to have an offspring with purple flowers we know we need either a homozygous dominant genotype or a heterozygous genotype, just anything that has a dominant allele that will overpower and give us that purple color. If we look at the Punnett square, we see actually all 4 genotypes in the Punnett square are heterozygous. So we've got 4 successes out of 4 possible genotypes, which gives us a 100% chance of the offspring having purple flowers. For problem 2, we're told that each parent is heterozygous, so once again to write out those genotypes, we've got a dominant allele along with a recessive allele for each parent. I pre-set up my Punnett square for this one just to be a little speedier. So again, let's write parent 1's across the top and parent 2's down the side. And let's go ahead and do our crossing procedure in order to find out every possible allele combination from these 2 parents. The first 3 follow-up questions are asking about the chances of every genotype category. So looking at 2a, we're asked about the chances of having a heterozygous genotype. We know we need 1 dominant allele and 1 recessive allele for that. So let's look to the Punnett square, and it looks like we've got 2 instances of a heterozygous genotype. So those 2 successes over the 4 possible genotypes in the Punnett square gives us a 50%. Moving on to the chances of a homozygous dominant genotype, we would need 2 dominant alleles in order to get that genotype, and it looks like there is 1 instance of that in the Punnett square, so we've got 1 over 4, which gives us 25%. Last but not least, we're asked about homozygous recessive genotypes, and we would need 2 recessive alleles for that, and once again, there's 1 instance in the Punnett square, and 1 over 4 gives us 25%. Now, it should make sense why, when we add up all of these percentages for the genotype categories, we get 100%, because these genotype categories cover every possible genotype that this offspring could have. Any offspring has to fit into one of these categories. And actually, you'll see this 50, 25, 25 breakdown for every cross you do with 2 heterozygous parents. You'll always have those 2 instances of a heterozygous offspring, 1 instance of a homozygous dominant offspring, and 1 instance of a homozygous recessive offspring. So that's just something to keep in handy in case you're ever asked down the line to do a Punnett square for 2 heterozygous parents. You could save yourself a lot of time. The next 2 questions problem 2 asks us are the chances of the dominant phenotype, or getting purple flowers, and the chances of getting the recessive phenotype, which we know is white flowers. So, what do we need to get purple flowers? Let's see, we know that we would need any genotype with a dominant allele, so that will be either the homozygous dominant genotype or the heterozygous genotype. There are 3 instances of all of these genotypes in the Punnett square out of the 4 possible genotypes, so we've got a 75% chance of getting that purple flowers dominant phenotype. Following a very similar procedure for the recessive phenotype, we know we need just those two recessive alleles. There's only one case of that, so we've got 1/4, which gives us 25%. Once again, if we add up these percentages, we'll get 100%, which should make sense because any offspring of these two parents can only be either dominant phenotyped and with purple flowers or recessively phenotyped with white flowers. I think I made up the word phenotyped, but still, that 100% covers every possible physical expression of the trait. For problem number 3, the final Punnett square problem, we are told that the two parents each have white flowers. Writing out those genotypes, we know that they must have 2 recessive alleles if they're displaying that recessive phenotype. So, with that written out, we can go ahead and start our Punnett square using the same process we've used for all of the past few problems. Now we are asked, what are the chances of white flowers? We know for an offspring to have white flowers, we need that homozygous recessive phenotype. We look to the Punnett square and we see that every single genotype in there is, in fact, homozygous recessive. So 4 successes out of 4 possible cases equals a 100% chance of white flowers. Similarly, we can answer the question of what are the chances of purple flowers. We can do what we did before and just count all instances of the required genotypes for purple flowers in the Punnett square, and we would get 0 successes out of 4 possible outcomes, being 0%. However, another way of solving this problem is to look at purple flowers as just non-white flowers. We know from problem number 2, what we just talked about, that the chances of an offspring having a dominant or a recessive phenotype is 100% because it can only have that dominant or recessive phenotype. So if we know the chances of any phenotype, and we just solved for the chances of just the white flower phenotype, we should be able to subtract the white flower phenotype chances from the any phenotype chances to get the chances of a non-white flower. And since we're only looking at 2 possible phenotypes, any non-white flower is the same as a purple flower. So if we go ahead and fill this in, we know there's a 100% chance of any phenotype. We just solved for the 100% chance of a white flower phenotype, and 100% minus 100% equals a 0% chance of the offspring having non-white or purple flowers. Either approach to this question is completely valid. I just wanted to show you another way in case you find this a little bit easier or quicker.
At this point, you have been given the tools to understand and predict the genetic possibilities from one generation to the next, from parents to children. This is fun and all, but genetics really starts to get interesting when we can look across many generations, from grandparents to grandchildren, from cousins to cousins, from aunts to nephews. In order to trace traits through families, scientists developed a special kind of family tree called a pedigree. A pedigree tracks a trait through multiple generations of a family and can sometimes be used to fill in missing information about a family member we might not have all the details on. Pedigrees can be applied in many different scenarios, from tracing diseases to dog breeding.
So I'm going to show you an example of how you can trace through a pedigree using this pedigree for polydactylism. If you're not familiar, polydactylism is a unique trait in which an organism expresses extra fingers or toes. This trait is actually near and dear to my heart because one of my small cats, named Ernie, is a polydactyl cat. So he actually has extra toes on every single one of his feet, which that in itself is pretty rare. But if you look at this video clip, and if you'll stop biting me and hitting me, you can see that on his front feet, it looks like he's got thumbs. On one of his front feet, he actually has 2 extra toes, not just 1 extra toe. But he's a little weirdo, but we love him. And there are actually a lot of other cats like Ernie who show this polydactyl trait. Oh, and just for reference, here's a shot of my other cat, Marie's feet. She's not polydactyl, so now you understand how massive Ernie's feet are compared to normal cat feet. And this pedigree that we're about to trace through is showing us an example of a little cat family tree where we see some instances of the polydactyl trait. But before we get started, we have to look at the key for the pedigree. Each and every pedigree comes with a key to make your life easier when trying to decode this series of filled-in circles and squares. So here we have the key for this pedigree. And the symbols on this key are pretty universal. You'll probably see the same symbols carried throughout other pedigrees when you're in biology. But a square denotes a male, a circle denotes a female. A filled-in circle or square indicates a family member that is affected with the trait in question. So in our case, a filled-in circle or square will mean that this cat is affected with the polydactyl trait and has some extra toes. On the right side of this key, I'll show you what some of the connections between the shapes mean on a pedigree. A horizontal line connecting a circle and a square is a mating or marriage line, depending if you're looking at humans or animals or whatnot. This line indicates that these two parents have mated to produce offspring. The vertical line that stems from the middle of the mating line is called the line of descent, because essentially you follow this line of descent to see all of the descendants of these two parents. From that line of descent, you'll see, in this case, a little horseshoe-shaped line, and that is the sibling line. Siblings are connected via this one horizontal line coming from the line of descent, and usually it has little vertical stems that come out of it that lead to each sibling. It's a bit confusing to explain verbally, but once you see me walk through the pedigree, I think you'll have a better understanding of it all. And lastly, on the right side here, we see Roman numerals, which are just the generation numbers. So the parents that are in line with that Roman numeral 1 are part of the first generation we're looking at, and then the children, or the offspring, in line with Roman numeral 2 are part of the second generation of this family. The polydactyl trait is what is known as an autosomal dominant trait. Autosomal means that this trait is not linked to the sex of a cat. So an autosomal dominant trait is a trait that only appears when there is a dominant allele present. This means that any cat with a homozygous dominant genotype or a heterozygous genotype will show this polydactyl trait. Not all traits are autosomal dominant. In fact, when we move on to the murder mystery, we'll look at a trait that's autosomal recessive, and that just means that the trait is only exhibited by people who have the homozygous recessive genotype. Now that we have the key for this pedigree, we can actually get to tracing it. And what I mean by tracing the pedigree is filling in all of the missing genotypes for these cats on this family tree. So, just as a reminder, we're looking at gene P for polydactylism, and the dominant allele, or capital P, does code for a polydactyl cat, and the recessive allele, a lowercase p, will code for a non-polydactyl cat, or a cat with the normal amount of fingers and toes. When I trace a pedigree, I like to start with all the information I know first. So, all of these not filled in circles and squares are cases of a cat that's not affected with the polydactyl trait. The key has told us this. So, if these cats are not affected with the trait, we know that they're exhibiting the recessive phenotype, which means that they must have a homozygous recessive genotype. It's easier to start with this kind of information first because we don't need to do any further investigation into the parents or offspring of these cats because the pedigree key has already told us what their genotypes are since they're not showing the polydactyl trait. I'm going to start by focusing on the right side of this pedigree because it looks like we have a little bit more genetic information. So looking at cat number 8, we see that it is homozygous recessive. One of its parents, cat number 3, is also homozygous recessive. However, since each cat can only pass on one allele for each trait, we know that its other parent, cat number 4, must also have this recessive allele. We can also see that cat number 4 is filled in, which means that it's exhibiting the polydactyl trait. Since the polydactyl trait is only exhibited when the dominant allele is present, we know that the other allele of cat number 4 must be a dominant one. Therefore, cat number 4's genotype is heterozygous. To me, it seemed most obvious to then focus on cat number 9. Once again, cat number 9 is filled in, so it has to have a dominant allele somewhere, and based on its two parents, we see that dominant allele must have come from cat number 4, because cat number 3 does not have a dominant allele whatsoever. If cat number 9 is inheriting a dominant allele from cat number 4, it must be inheriting a recessive allele from cat number 3, because cat number 3 has nothing else to offer it. Now we can start to focus on the left side of this family tree. I start with cat number 7 because that's closest to a lot of the unknown cats that I have. And if cat number 7 is a child of cat number 1 and cat number 2, it must have inherited both of its recessive alleles from cat number 1 and cat number 2 respectively. Therefore, I know that cat number 1 and cat number 2 each have a recessive allele. Once again, we see that both cat number 1 and cat number 2 are filled in, which means that they're polydactyl cats, so they must have that dominant polydactyl allele. Therefore, both cat number 1 and cat number 2 are heterozygous cats. Now, the only two cats we have left to figure out are cats number 5 and 6. They're each filled in, so they must each have at least one dominant allele. We can try to refer to their parents for what their other allele might be, but when we do so, we can see that no matter which parent has given them that known dominant allele, the other parent could give them either a dominant allele or a recessive allele. That's actually all the information that we can figure out about cats 5 and 6. They can either be homozygous dominant if they get a dominant allele from each parent, or they can be heterozygous if they get a dominant allele from one parent and a recessive allele from another parent. Sometimes with pedigrees, this will be the case where you can't know the exact genotype of every family member that you look at, but we want to get as close as we can get. So even the fact that we've narrowed down that these cats are either homozygous dominant or heterozygous is us discovering something that we may not have known before. And that is an example of how you might trace through a pedigree.
Genetics can tell us a lot more than just how a person looks. Did you know that genetics are incredibly important in determining blood type? Just like eye color, blood types have dominant and recessive alleles.
Here, I'm going to talk a little bit about the genetics behind blood types because you'll need this information when you move on to the final activity of solving the murder mystery. There are 4 families of blood types, and you may have heard of them before. There's type A, type B, type O, and type AB blood. In order to exhibit type A blood, a person must have either 2 dominant A alleles or a dominant A allele and a recessive O allele. A recessive O allele is just indicated with a lowercase I. In order for a person to have type B blood, they can either have 2 dominant B alleles or a dominant B allele and a recessive O allele. Pretty similar to type A blood. In order for a person to have type O blood, they have to have 2 recessive O alleles. This is the only way that they can have this blood type. Type AB blood is very unique. Instead of having 1 dominant and 1 recessive allele, people with type AB blood actually have 2 dominant alleles, 1 dominant A allele and 1 dominant B allele. This phenomenon is called codominance. If 2 alleles are codominant, that means that they will both be expressed rather than one of them being masked up like a recessive allele. These are the 4 main families of blood types. You may have also heard of blood types like A positive or O negative, and the positive and negative aspect of blood types actually is due to another gene. To simplify things a bit, I'm just going to be focusing on blood type family today and not positive and negative blood.
The time has come for you to put your newfound genetic skills to the test and solve a murder mystery. You will have to use your knowledge of dominant and recessive alleles, of Mendel's 3 laws of inheritance, of Punnett squares, and of tracing pedigrees in order to stop this cold-blooded killer in their tracks. It is up to you to bring justice to our victim, Monsieur Mendel, and save the rest of the Mendel family. Are you up to the task? Of course you are. Let's get started.
It was a dark and stormy night at Mendel Manor. The wind was so furious that the shutters were slamming to and fro. The thunder shook the grounds of the manor like an earthquake. Lightning pierced the air and created images of shattered sky. The storm did not seem out of the ordinary. In fact, the weather was never too kind around Mendel Manor. Suddenly, the sound of a vase shattering woke Madame Mendel, the matriarch of the household. She silently crept down the stairs to the room that held the Mendels' most prized possessions, the gallery. Madame Mendel was horrified at what she saw by the dim light of her candle. There, on the floor, lay her husband of 45 years, Monsieur Mendel, bleeding from a large gash on his head. There was shattered vase spread all across the floor, as if it had been used as a weapon. Madame Mendel let out a shriek that startled the other members of Mendel Manor from their chambers. The remaining members of the house ran into the gallery, and most were too astonished to utter a word. Sir Marcus was the only one who was able to open his mouth and dial 911 to summon the police. While there were many questions surrounding what exactly had happened that stormy night, one thing was certain. This was a cold-blooded murder. Once the police arrive on the scene, they begin to gather evidence. A blood sample is taken from Monsieur Mendel, as well as fingerprints from the vase, which the police have confirmed is the murder weapon. The police also acknowledge that nobody broke into the house that night, as they found no footprints or tire tracks on the muddy drive. The murderer was a member of the Mendel household. Can you use the police evidence and family history to solve the murder and bring justice to Monsieur Mendel? Pause the video and complete the murder mystery packet to find out. Thanks to your hard work and your superior genetic skills, you were able to help police identify the true murderer of Monsieur Mendel. The only person who could have possibly left behind type B blood and heterozygous fingerprints was Miss Maisie! Miss Maisie thought that by offing Monsieur Mendel, she might have a chance at his billion-dollar fortune. Unfortunately for her, DNA doesn't lie. But how exactly were you able to crack this case wide open? Let's review some of the pedigrees and see how you got to your results. I'm not going to voice over every solution inside of these pedigrees. However, I will show you my work when I went to solve the pedigrees so that you can get an idea of my thought process. I used a very similar process to the polydactyl pedigree walkthrough, so you can go back to that tutorial as well if you want a more specific description of how you go about tracing a pedigree.
Were you able to find the murderer? Whether or not you cracked the code, I hope you had some fun putting your genetic skills on the line. I am really impressed with how much you learned about DNA and genetics today. Let's take a look back on some of the important topics that we covered.
First, we talked about Mendel's 3 fundamental laws of inheritance. Then, we dove a little deeper into the differences between dominant and recessive alleles. We even touched on sexual versus asexual reproduction, and then moved on to a large portion of the module learning how to draw and use Punnett squares. We learned how to trace through pedigrees and fill in some missing genotypes, and even learned a little bit more about blood typing.
Now you have more of an understanding of why the human population is so diverse. Genetics are everywhere and contribute to both the major and minor differences between all organisms on this planet. To this day, scientists are learning more about genetics and are finding really incredible applications, especially with regards to genetic engineering. Scientists are learning how to manipulate DNA sequences in order to create new medicines and cures, and also do things like create a drought-resistant crop. The Human Genome Project is another interesting scientific study that aims to identify, map, and analyze every single gene in the human genome, which is every gene found in human cells. There are about 40,000 genes, give or take. If you're interested in learning more about DNA-related research going on at Carnegie Mellon University, make sure to check out the virtual tour of Dr. Rebecca Taylor's Microsystems and Mechanobiology Lab, which is also available on the Leonard Gelfand website. Thanks so much for watching, everyone, and I hope that you learned something new today, or a few new things. If you liked this video, make sure to check out some of the other educational videos that my classmates have made. They cover a whole range of topics, not just within the realm of biology. Once again, I'm Shaylin, and this has been Discovering DNA.